Alrighty, welcome to this episode of Lectures with Liddington. Today we're going to continue talking about a particular component of the cell cycle. Last episode we talked about the cell cycle and these different phases that a cell goes through over its lifetime between divisions. Today we're going to specifically talk about that division, and that division is mitosis. So let's go back and sort of revisit these phases. So if we break it up in general, interphase is the longest section of the cell cycle. The cell spends the most time here in interphase. Then it goes through a cell division. This division here is called mitosis. And then it all ends in what we call cytokinesis. And this is when the two cells actually pinch off and we have two new daughter cells. Remember that vocabulary word, daughter cells, those are the new ones. But here is our focus for today. We're going to talk about what happens when a cell divides, and that process is called mitosis. So in last episode, you heard the abbreviation PMAT a couple of times, and that is a nice little mnemonic that we use to help remember the phases of mitosis, and that's PMAT for prophase, metaphase, anaphase, and telophase. So what does that look like? Let's look at some visual representations of what's going on in each of these phases. So interphase, this is in between. Inter is in between. This is in between cell divisions. The cell is doing its job. It replicates DNA. This is where we go through that G1, S, G2 phases that we talked about last episode. And here in interphase, because a cell spends most of its time here, most of the cells we look at, if we're looking at cells under a microscope, most of them are going to be seen in interphase. So notice here, this is our nucleus, and we have this like mess of dots. That mess of stuff, that's called chromatin. That's the form that DNA takes when we're in interphase. It's just all strewn about because we need to be able to access that information if we're going to be able to replicate that DNA. But once we start moving into cell division and we move into mitosis, it just doesn't make sense to have our DNA in this form of chromatin. It's going to be really difficult to divide everything out evenly. That's one of our goals in cell division, is to evenly divide all that genetic information into each of those new daughter cells so that they all have a full set of the information. So here in prophase, this is our first phase here. Our DNA starts to form chromosomes. This is where we take that whole mess of chromatin and we wind it up really nice and neatly. So this is what it looks like in a cell, but down here is a nice illustrated version. This is where we get those nice neat little X's. We take all of that chromatin and we wind it up. Some of the other things that happen here in prophase, so P meaning prepare. This is where we're preparing that cell to divide out. So our nuclear membrane here around the outside of our nucleus starts to degrade, right? We need to get that out of the way before we can start moving those chromosomes different places. And then a couple of structures here that will form. In animal cells here, these are centrioles. These are necessary in order to anchor what are called spindle fibers. So these little things here are called spindle fibers, and those are going to form in order to sort of almost like bungee cords that are going to hook onto our chromosomes and pull them, but we need to make them first. So this phase here in prophase, where we're preparing, we form those spindle fibers and we form centrioles in animal cells if they're needed. The next thing, once we sort of get prepared here, we make those spindle fibers and centrioles if needed. We take our DNA, wrap them up nice into chromosomes, and we start breaking down our nuclear membrane. Then we make it to M, metaphase. M for middle. Now this is a great little mnemonic because here in metaphase, they literally line up at the middle. So notice here in our actual cell, right there are all of our chromosomes lined up right at the middle in our diagram. It's just nice and neat and pretty. But in our actual cell, it's pretty neat, right? Those chromosomes line up right in the middle. Those spindle fibers that we formed before, they hook on to what are called the centromeres of the chromosomes. Those are these middle points right here. Notice in our little diagram, those spindle fibers are all hooked up right at the center of the chromosomes. Those are the centromeres. So they hook on, they latch on, it's almost like a belt, right? They hook on right there. And then in our animal cells, they will be anchored over here at our centrioles. In plant cells, they have that extra layer, that cell wall, that sort of helps in this process. But in our animal cells, we need those anchor points for the spindle fibers. So the spindle fibers hook on at the centromeres, and our chromosomes all line up in the middle. Next, anaphase, A for away. So those spindle fibers will pull those sides, the two sides of those chromosomes apart. Each side of a chromosome is called a chromatid. So if I go in here, this is one chromatid. It has enough genetic information for that cell. So this has a full set of genetic information. So here, 1, 2, 3, 4, there's 4 chromosomes, each with a full set of information. So the cell, the daughter cell that I'm going to get here, will have everything it needs informationally because we already replicated the DNA. So we doubled it before, and now we'll have a single set here and a single set here. So each chromatid gets pulled away from each other by those spindle fibers here. Now, telophase. Telo means terminal. So this would be the last phase of mitosis. At this point, we're sort of undoing all the things we did during prophase because now we're preparing those two almost new cells to become new cells. So we'll start to lose those spindle fibers. So notice you're not seeing any spindle fibers here. Well, that's because they start to degrade. We don't need them anymore. Notice these dotted lines. That's because my nuclear membrane is starting to come back. I'm starting to rewrap those chromosomes into nice, neat little packages so that each of those new cells that will be a result of this process will have a new nucleus that contains its set of DNA here and its set of DNA here. And then the other thing that happens, notice how in our actual cell here, it's not as neat anymore, right? It's starting to unwind. So my chromosomes that used to be all nice and neat are starting to get all smushed out into chromatin. It's starting to look like that spaghetti mess again. Not quite there, but it's starting to get to that point. So my chromatin starts to reform, I lose my spindle fibers, I start to gain back my nuclear membrane, and my two cells are starting to split off. In plant cells, I get what's called a cell plate. That cell wall that's in between is what is going to start that division process. In animal cells, what forms here in the center is called a cleavage furrow. It's going to look a little bit different because those animal cells, they don't have that cell wall, whereas plants do. But it's the same general idea that I get a piece in the center that says, this is where we're going to split off. Lastly, we end here with cytokinesis, and this is the point at which I snip into 2 new daughter cells. I started with 1 parent cell. Now at cytokinesis, I officially have 2 daughter cells, and these 2 daughter cells can now go into G1 phase of their own cell cycles. So let's pause here. Do a little practice.